Fermentation and anaerobic respiration. Organisms have methods for obtaining energy from fuel without the use of oxygen. This is a giant fermenter, which makes lactic acid by harnessing the catabolic activity of microorganisms through the fermentation of mild cornstarch. This fermentation occurs on such a large scale that the plant can produce 140,000 tons of polylactic acid, PLA, derived polymers, which are used to make biodegradable implants and packaging. Topics covered in this module: catabolizing fuel without oxygen, anaerobic respiration using terminal electron acceptors other than oxygen. Fermentation allows glycolysis to continue in the absence of terminal electron acceptors. Major objectives of this module: distinguish between obligate anaerobes and facultative anaerobes. Give examples of fermentation, and compare anaerobic respiration and fermentation. 5 pages left in this module. Humans have been making beer, wine, and other alcoholic beverages for nearly 10,000 years. Only in a more recent time has the process of making alcohol become more fully understood. Sugars dissolved in water or fruit juices are converted to alcohol and carbon dioxide. The carbon dioxide forms bubbles in the fermented product, making the liquid appear as though it is boiling. The appearance accounts for the name fermentation, which comes from the Latin word fervere, meaning to boil. But how is fermentation linked to living cellular processes? All cells perform cellular respiration, in which the chemical energy in molecules ranging from glucose to methane to hydrogen is released for use in various cellular functions, including movement, growth, and reproduction. Invariably, the released energy is passed through an intermediate compound, adenosine triphosphate, before expended. We are most familiar with plants and animals that use aerobic cellular respiration to obtain energy, but many organisms use alternative processes that do not require oxygen. These organisms utilize different types of cellular respiration depending on the environments to which they live. Nevertheless, cellular respiration of some kind is vital for all forms of life on Earth. Catabolizing fuel without oxygen. Cellular respiration involves reduction oxidation, redox reaction, in which organic molecules such as glucose are oxidized through removal of electrons, while other molecules are reduced by addition of electrons. All known forms of cellular respiration begin with glycolysis, in which glucose is oxidized in the organic compound pyruvate. Glycose itself does not require the presence of oxygen. Based on the comparative studies and genetic sequence data, glycolysis appears to have emerged very early in the evolution of life as a way to generate ATP, at a time when there was little free oxygen in the environment. Following the advent of photosynthesis and the release of free oxygen as a byproduct, atmospheric oxygen eventually reached levels that favored the evolution and emergence of organisms that used aerobic respiration. Habitats with little or no oxygen still exist, such as in certain soils, in mammalian intestines, and around underwater hydrothermal vents. Figure 1. When oxygen is limiting or absent, organisms, almost all of them microorganisms, use fermentation and anaerobic respiration to generate ATP. Chemolytrophs, organisms that use inorganic material for energy, survive at these hydrothermal vents by oxidizing hydrogen sulfate and reducing O2. They are part of a complex ecosystem that includes sulfate reducers and methogens. Table 1. How do organisms such as these generate energy when there is little or no oxygen? Figure 1. A hydrothermal vent in the ocean floor. Chemosynthetic anaerobic bacteria thrive in these volcanic vents, which are rich in oxygen, sulfate, and dissolved minerals, but low in oxygen. And this is a list of electron donors and acceptors that show the variety of molecules used by different types of organisms. Anaerobic respiration using thermal electron acceptors other than oxygen. In organisms that perform aerobic cellular respiration, oxygen is a thermal acceptor of the electrons from pyruvate oxidation. Oxygen works well as the terminal electron acceptor because of its high reduction potential or electronegativity. However, the absence of oxygen, other substances serve as a thermal electron acceptor for the redox reaction of cellular respiration in processes collectively known as anaerobic respiration. Like aerobic respiration, anaerobic respiration Like aerobic respiration, anaerobic respiration uses an electron transport chain to oxidize NADH and FADH2 back to NAD plus and FAD. However, anaerobic organisms have specialized enzymes that allow substances such as carbon dioxide, nitrate, iron, and sulfate to serve as a thermal electron acceptor at the end of an electron transport chain. Table 1. Because these molecules are not as electronegative as O2, they cannot extract as much energy from glucose oxidation as aerobic respiration can produce. Indeed, anaerobic respiration yields only about 75% as much ATP as does aerobic respiration. All respiration processes begin with glycolysis, but depend on the environment conditions at the specific biochemical processes available to the cell. Pyruvate can be diverted to oxidative phosphorylation, anaerobic respiration, or fermentation. Fermentation allows glycolysis to continue in the absence of terminal electron acceptors. In fermentation, molecules produced during glycolysis also act as electron acceptors. Glycolysis converts one molecule of glucose into two molecules of pyruvate, leaving two molecules of ATP in the process. During glycolysis, NADH is reduced to NAD plus is reduced to NADH as glucose is oxidized. When neither oxygen nor another terminal electron acceptor is available so that ATP can be produced using an electron transport chain and chemiosmosis, then NADH must be recycled to NAD plus through some other means. If NAD plus is not regenerated, then glycolysis and ATP production will grind to a halt. Fermentation couples the oxidation of NADH to NAD plus with the production of pyruvate. Fermentation allows glycolysis to continue uninterrupted by regenerating NAD plus, which in turn can accept more electrons from the reactions of glycolysis. The steps of fermentation following glycolysis do not yield ATP, but are necessary to sustain the production of ATP from glycolysis. Of the three forms of cellular respiration, fermentation, anaerobic respiration, and aerobic respiration, fermentation is the least efficient, producing only about 7% of the ATP produced by aerobic respiration. There are numerous fermentation pathways used by microorganisms whose products are often exploited by humans to produce alcohol, bread, and cheese, fermented foods, and industrial solvents. One of the pathways to lactic acid fermentation involves the oxidation of NADH to NAD plus is coupled by the reaction of pyruvate to lactic acid. Lactic acid fermentation is a one-step process. Pyruvate is directly reduced to lactic acid. Lactic acid fermentation in bacteria is commercially important to the dairy industry for making cheese and yogurt. In animal cells, most notably muscle fibers, lactic acid fermentation also occurs during periods of intense exercise. Muscle fibers normally use aerobic respiration to produce ATP, but very active muscles may outpace the speed at which oxidative phosphorylation can produce ATP. Glycolysis generates ATP more rapidly, so when the quick bursts of energy are needed, fermentation allows ATP to be made more quickly, albeit inefficiency and the expense of lactic acid accumulation. The lactic acid produced by muscle fibers is eventually circulated to the liver, where it is then converted to pyruvate and can be used in aerobic respiration. Figure 3 shows the lactic acid and alcoholic fermentations. In lactic acid fermentation, the two pyruvate molecules produced during glycolysis are reduced to two molecules of lactic acid while simultaneously oxidizing NADH back to NAD+. B. To produce ethanol, the two pyruvate molecules produced during glycolysis are first converted into two molecules of acetaldehyde, releasing carbon dioxide to the process. Next, acetaldehyde molecules are reduced to two molecules of ethanol, while NADH is oxidized back into the NAD+. Alcoholic fermentation converts pyruvate to ethanol and carbon dioxide in a two-step process. First, the 3-carbon molecule of pyruvate is converted into a dual-carbon molecule of acetaldehyde, releasing a molecule of carbon dioxide in the process. The acetaldehyde is reduced to ethanol, while NADH is oxidized back into NAD+, which can be reused in glycolysis to produce more pyruvate. Figure 3b. The fungus Saccharomyces cerevisiae is a common species of yeast used by beer brewers, winemakers, and bread makers, whose products are driven by alcoholic fermentation. In the closed aqueous environment of what becomes beer or wine, the yeast are available to oxygen to grow. Once the oxygen has been depleted, the yeast continue to metabolize the sugar that is present. Without oxygen, the yeast ferment pyruvate to acetaldehyde and eventually alcohol to recover the reduced agent NADH plus required for further glycolysis. This results in accumulation of waste products, carbon dioxide, and ethanol. The carbon dioxide forms bubbles in beers and champagne that makes bread dough rise. In bread, most of the ethanol evaporates during the baking process. Current research contradicts the view of lactic acid as a useless waste product. Researchers are trying to better understand the role of lactic acid in muscles. George Brooks, a professor at the University of California at Berkeley, is challenging the conventional wisdom that lactic acid is purely a waste product of intense exercise, a belief commonly led by marathon runners or other endurance athletes. Through research, Brooks determined that mitochondria could metabolize lactic acid as a secondary fuel source, thereby potentially producing more energy and preventing further accumulation of lactic acid. An improved understanding of lactic and metabolism could lead ways to make more ATP available for muscle contractions and delay fatigue. Why does fermentation produce less ATP than respiration does? Fermentation yields less ATP than aerobic respiration because the end products of fermentation still contain most of the stored energy of the starting material. Table 2 compares the energy of combustion measured as alpha-B for heat, glucose, lactic acid, and ethanol. The two molecules of lactic acid produced during lactic acid fermentation contain 94% of the chemical energy contained in the input molecule of glucose, while the two molecules of ethanol produced during alcoholic fermentation contain 95% of the glucose molecule's energy. However, the chemical energy stored in lactic acid and ethanol cannot be harvested without the availability to further oxidize these fermentation end products using the citric acid cycle and some version of the electron transport chain. Thus, these end products are exerted from the cell, which is often used for humans who use these compounds in food production and industry. Table 2 comparison of the amount of energy in the glucose and its fermentation products. 1. The first law of thermodynamics states that the energy cannot be created or destroyed, it can only change forms. The alpha-B or combustion for both ethanol and lactic acid reveals how much potential energy the end products of fermentation still contain. This column indicates the number of molecules formed per molecule of glucose oxidized in glycolysis. Bioskill. Measuring fermentation rates. One way to measure the rate of fermentation of a culture of microorganisms is to measure the change in pH of the culture medium because acids are often a product of fermentation. As the fermentation process produces more and more lactic acid, for example, the medium becomes more acidic and the pH decreases. A probe is used to constantly monitor the fermentation process, and the pH is plotted as a function of time. Once all of the sugars are consumed, the microorganisms eventually die and the pH will stop changing. Figure 4 is measuring lactic acid and fermentation by pH. The pH of a solution containing bacteria undergo lactic acid fermentation is plotted as a function of time. The pH drops from 5 to 4 in 205 hours, or 8.5 days, due to the accumulation of lactic acid. A second way to measure the rate of fermentation is by monitoring the amount of gas produced. Some, but not all, fermentation produce gas like CO2 or H2. Alcoholic fermentation releases CO2 as a byproduct. Thus, the rate of fermentation can be calculated as a function of the carbon dioxide concentration of the culture medium. Here again, once all of the available sugars are consumed, further release of the carbon dioxide ceases, signaling the end of the fermentation. Some organisms prefer oxygen and some avoid it. Some organisms or cells require oxygen because oxygen is the only terminal electron acceptor that they can use. Such organisms are known as obligate anaerobes. In contrast, some organisms are actually killed by the presence of O2. Such organisms are obligate anaerobes. Lack of necessary enzymes to degrade certain toxic byproducts of oxygen. Other organisms are facultative anaerobes, meaning that they can survive in the presence or absence of oxygen. Facultative anaerobes typically employ anaerobic respiration when oxygen is available, but have other pathways such as anaerobic respiration or fermentation available when oxygen is absent. The preference for different oxygen environments by different species of bacteria can be observed experimentally. When a growth in a liquid media in a test tube, obligative anaerobic bacteria gather near the surface of the media to maximize their exposure to oxygen. In contrast, obligate anaerobic bacteria cluster at the bottom of the test tube in order to avoid oxygen. Facultative aerobic bacteria can be found along the test tube since they can tolerate the absence of oxygen. However, more of these bacteria gather near the surface because aerobic respiration produces more ATP and thus preferred over either anaerobic respiration or fermentation. Bacteria with the different requirements of oxygen distribute themselves differently in a liquid media and a test tube. Obligative aerobic bacteria are found nearest to the surface of the media where oxygen concentration is the highest. Obligate anaerobic bacteria move to the bottom of the test tube to avoid oxygen. Facultative anaerobic bacteria can be found along the length of the test tube, but often prefer the region close to the surface because of the increased efficiency of ATP generation when oxygen is available. Summary. Compare anaerobic respiration and fermentation. Where oxygen is lacking or absent of cellular respiration, some organisms can use sulfate, nitrate, carbon dioxide, oxygen, iron, and other molecules such as the terminal electron acceptor in the electron transport chain. Linked to chemiosmosis, these thermal electron acceptors are available to generate a fair amount of ATP by anaerobic respiration, albeit producing only about 75% as much ATP as is produced by aerobic respiration. In fermentation, organic molecules such as pyruvate and acetaldehyde serve as electron acceptors so that NADH can be oxidized back into NAD+, which allows glycolysis to continue uninterrupted by maintaining a steady supply of NAD+. Fermentation itself does not produce additional ATP. Objective. Give examples of fermentation. Two well-studied fermentation pathways are lactic acid fermentation and alcoholic fermentation. In lactic acid fermentation, pyruvate is directly reduced to lactic acid. In alcoholic fermentation, pyruvate is first oxidized into acetaldehyde, and acetaldehyde is then reduced to ethanol. In both of these processes, NADH is simultaneously oxidized back into NAD+, freeing it to accept more electrons from the reactions of a glycolysis. Objective. Distinguish between obligative anaerobes and facultative anaerobes. Obligative anaerobes are killed in the presence of molecular oxygen because they lack the enzymes needed to inactivate toxic byproducts of oxygen. Such organisms depend on anaerobic respiration or fermentation for energy generation. Facultative anaerobes are organisms that can survive in either the presence or absence of oxygen. In these organisms, both aerobic and anaerobic respiration pathways are available. However, because aerobic respiration is often more efficient at ATP generation than either anaerobic respiration or fermentation, aerobic respiration is often preferred when oxygen is available. Key terms. Alcoholic fermentation. The form of fermentation in which ethanol is produced as a product of NADH oxidation. Anaerobic respiration. The form of cellular respiration in which substances other than molecular oxygen such as carbon dioxide, sulfate, iron, or nitrate are used as the terminal electron acceptor. Chemolotroph. An organism that oxidizes inorganic compounds to yield energy. Facultative anaerobe. An organism that can live in either an anaerobic or an anaerobic environment. Fermentation. A biochemical pathway that allows NAD+ to be generated from NADH after glycolysis when the electron transport chain is not available. Known pathways include lactic acid fermentation and alcoholic fermentation. Glycolysis, the biomedical process by which glucose is broken down into two molecules of the three-carbon compound pyruvate. Lactic acid fermentation, the form of fermentation in which lactic acid is produced as a product of NADH oxidation. Obligative anaerobe, an organism that can survive and thrive only in the presence of oxygen. Obligate anaerobe, an organism that cannot survive in the presence of oxygen. An oxygen environment may be too toxic to some members of this group.